because somehow people think that this is symbolic. I think while it's symbolic, we should all realize that this is one day where we at least reaffirm our beliefs and change our attitude towards systems that really make society equitable. I think by saying we will empower women, actually, that's not the right word. That's my belief, because I don't think anybody is worthy enough to empower anyone else, period. Definitely not a woman, according to me. So it's about making more equitable place. And in that context, I think institutions like ours have a huge mandate. And I said this in CDFT as well and many other platforms, is that it's really sad that the maximum number of our PhD graduates are actually girl students. And yet, the number of people who go on to do full-time careers in science is very limited, right? And often we are given an excuse saying that, oh, we don't find people. It's true. It's quite likely that we don't find enough good women scientists, but that is also possibly partly an excuse. I think we need to do more, and this is my request to all our men faculty colleagues and the administration to say that we should do our best to make sure that we bring this back. But then that also happens only because probably we are failing in our ability to foster and support careers of especially women after they finish their PhD. So I think there is a huge need to do this. And this is something that all of us should actively work on. Only then the pipeline gets completed. Otherwise, we will not complete the pipeline. Now, maybe some of you have heard this story because it's something very personal. But I will tell you how when one woman changes, it changes multiple generations. It's my own family story. My grandmother was barely educated. Maybe she didn't even go to school beyond 4th standard. She was in a village near Mysore. She didn't herself know much to write or read. She had 7 daughters and 4 sons. And she took a vow that she will educate all her daughters. This is in 1920s and 1930s. Okay, much before independence. Much before anybody even thought that their girls should even do 10th. My mother and all my aunts did minimum post-graduation in 1940s and 1950s. Okay. Now, maybe you would have heard about this story by many people. It's true. There are many such stories. But what really struck me when I heard it for the first time when I was young is that apparently my grandmother broke ties with her own mother and with her sister who opposed girl education. She said, hell with my mother, hell with my sister, I will not keep in touch with them. So my mother never remembers her grandmother or her aunt, because my grandmother said that anybody who becomes a stumbling block in educating my daughters is not my relative. I'm just wondering, who do you call empowered now? I would say my grandmother was more empowered than my mother, although my mother retired at a very high position in a government job. So empowerment is not necessarily getting a job and earning money. Empowerment is to make sure that you actually make a change. And I'll tell you, if you stand up, you will become an example for many people to change. Okay, that's my belief. And we were all brought up in a household where, in fact, men were made to feel less than women because we were less than women. It's not that we were made to feel. So I think that's the spirit I would like to really request. And although today we have Dr. Rashna as our speaker, this is so personal to me. And every day when I wake up, I think about what I would have been if I didn't have this influence. I'm not joking. Almost every day it crosses your mind, right? Because you are what you are based on your upbringing. This is very important. And I strongly believe that if girls and students who are now doing PhDs, if you stand up and if you say, I will take that slightly harsh stance that I matter and what I think matters for society. I think we'll do a change. So with that, I'll stop here. But like I said, I would have really loved to see more participation, and this is important. And I request the administration and the faculties to do one thing. I've told this in CDFT as well, is that one of the ways to tackle this is also about, although when we come here, we all assume we are equal. And as the director, I would like to request one thing, is that let's have a workshop. And I'm going to do this in CDFT. So hopefully before I leave from here, I would like to see that workshop. Is that to see how many of us truly have potential biases against women. When I say against women, it's not about against women. We have biases about what women can or cannot do, or what men should or should not do. Okay. Sometimes women themselves have biases. Okay. I again gave a story about Dr. Shobana, who was in JNC. I don't know if you have heard about this. So when she was sitting about 10 years ago in an aircraft, and that was initially when women pilots were commissioned, commercial aircraft. Apparently, Dr. Shobana herself felt from JNC that when the captain was a woman, she said, "Oh my gosh, a woman is flying glove flight." See, it's not biases only by men, biases by women, thinking that women can't do. Right? So I think we should have a workshop to actually help ourselves identify what explicit and implicit biases we have. Some biases are explicit. Some biases are implicit. That means I myself may not know that I have a bias. Right? So workshop will help address this. Okay, sometimes we may not be able to change. Maybe sometimes, it's very difficult to change, but it's good to recognize whether you have biases. So I really request Harjit and RSC to take this up more actively, and Madhuri, if you can get this done. We should have a workshop, and this workshop happen almost every year, because this is as important as any other activity that we do. With that, I'll stop here. Thanks, Rashna, for coming. And of course, thanks all of you to make sure that this is an important day. Thank you.
Thank you, Professor Vilas. You have already given to page. Now I would like to invite Professor.
Good afternoon, everyone. Wish you all a very happy International Women's Day today. As you all know, that our guest today is Dr. Rasna Bhandari from CDFD. And it's my honor to introduce her in front of everybody here. Dr. Bhandari obtained her master's and PhD degree in biological sciences from Indian Institute of Sciences, Bangalore. And in 2001, she moved to University of California, Berkeley for the postdoc fellowship. And she worked there on structural biology, biochemistry of proteins involved in cell signaling. In 2003, she joined Johns Hopkins School of Medicine in Baltimore, where she worked on deciphering the role of inositol pyrophosphates as signaling molecules. In January 2008, she joined CDFT as a staff scientist, where she continues to pursue her study on signal transduction in biological systems. She has been the recipient of DBT Wellcome Trust Senior Fellowship, and she's also a National Academy Sciences Fellow. And the list is very long for the accomplishments and awards, but I'll be very brief here. With this brief introduction, I request Dr. Bhandari to deliver her lecture on Give to Gain, Empower to Rise.
This is your microphone. Very clever. Turn it off. Yeah, can you hear me? Oh, wonderful. And this also has a pointer? Okay, great. All right, very thoughtful that I don't have to hold three things. Sometimes it's a forwarder, pointer, and that microphone thingy. So, one step towards making it easier for women who don't carry pockets with them all the time. Thank you. I just put this slide up here so that you could take a look at it as to what the theme is this year on International Women's Day. And I also think I'll do a quick show of hands to gauge who's sitting here. So I see some of our friends there. Hi. I can talk in Hindi if it helps. हिंदी आपको समझ में आती है? Okay. थोड़ा ट्राई करूंगी हिंदी में बोलने का तेलुगु रादू sorry ನನಗೆ ಕನ್ನಡ ಬರುತ್ತೆ but I don't think anyone speaks Kannada here. By the time I reached Hyderabad, my brain was done learning languages. PhD students, please raise your hands. All right, out in full force. And if I'm just looking at it correctly, I am seeing more than 50% women in the PhD students here. Am I correct? Yes? All right. Faculty colleagues, few here and there. Thank you. 50% women, right, Madhuri? So that's what Ullas was talking about, the journey with the leaky pipeline. But that's not what I'm here to talk about today. I'm just here to share my own story, because when I discussed what I'll be saying today with my husband, he said, don't go give gyan, just talk about yourself. So that's what I plan to do, although I don't usually like talking about myself. But I think in one does this, your own reflection helps you realize where you are, what you've done, where you go, and how you can make a difference. Is this echo bothering you? You're okay with it? Okay, you're used to it. All right. And who else? Admin, support staff, finance. All right, we have purchase, admin, and support staff, and one more hand there. All right, thank you for coming out today. Thank you for making the time. Anyone? Why do we celebrate International Women's Day? क्यों मनाते हैं हम ये दिवस? International Women's Day. क्यों मनाते हैं हम? Are you banned from using Google here in the room? You can look it up and tell me. Okay, whenever you're ready with the answer, let me know. क्यों मनाते हैं और क्यों मनाना चाहिए? Why is it important for us to mark this occasion? वो तो बता सकते हो बिना गूगल के. Anyone? No, don't have the fastest internet over here. And why should we mark this day? जरूरी है क्या ये ऐसा महिला दिवस मनाने के लिए? क्यों मनाते हैं हम? आपने रंगोली तो बहुत अच्छी बनाई, क्यों मनाते हैं हम ये दिवस? क्योंकि डायरेक्टर ने बोला कि बैठने के लिए यहाँ ऑडियंस में. All right, my male colleagues, why do we celebrate International Women's Day? Please, whom do I put on a spot here? Vapaditya. Why? Vapaditya is an old friend, he won't mind.
So you see, for very long in the Indian society, women has been not given the right position. By calling right position means not the equal position in the society. It has been very long patriarchal society. Although in many places there are certain changes has come, in general it is more or less patriarchal, where women in the family, start from family, so has been seen as lesser than boys or the men. It is, and as we progress over as a human and human society progress, we realize that, and it is biological as well as evolutionary, that women are no less than, rather than it is in the higher side of the gender. And it is high time we give them the right opportunity. It has been suppressed many times, not by women's choice, but the societal choice, which are mostly men.
Perfect.
Give them the right positions. and the importance of women in our life, society, as well for the progress of the country and the globe per se, which is not possible
Without 50% of the
it's
population.
extremely important, celebrating women and promoting.
All right. Who here is looking forward to a day when we don't need to have International Women's Day? Yeah. Someday, maybe not in our lifetime, maybe in your children's lifetime, maybe in your children's lifetime, we will not need to mark this occasion, because those inequities will cease to exist. हम चाहते हैं कि ये Women's Day हो ही ना, औरतें सचमुच मर्दों के बराबर ही रहें, इतना बराबर रहें कि ये हमें सोचने की जरूरत ही ना पड़े कि औरतों के लिए और उनके कंट्रीब्यूशन, उनके इक्वालिटी के लिए एक दिवस दिया जाए. But we are not there yet. So the theme this year is give to gain. Give to gain, reciprocity and support. What you give, you will gain back manyfold. When women thrive, we all rise. Giving is not a subtraction, it's an intentional multiplication. आज तक देने से कोई गरीब नहीं हुआ. If you give, you will receive manyfold. Through donation, knowledge, resources, infrastructure, visibility, advocacy, education, training, mentoring, or just give your time and that, or give your thought. Pause and think. Give that much time to think, reflect, and that giving is going to help make our society more equitable. So, what will you give to gain gender equality? This is a moment to reflect on that. Does anyone have the answer? Who had the fastest Google answer? Come on, I'm sure you all did. Labor rights? Labor rights and voting rights in New York, labor rights in Russia. Okay, so voting, women's suffrage, women were not allowed to vote for the very longest time. So 8th of March marked that day in multiple places for different things. And then the UN selected it as International Women's Day. And I forget which year it was selected by the UN. This is what I came up with when Madhuri wrote to me, give to gain. That's the theme this year. So then I looked it up and I thought a lot about it and try to make it personal, my journey. If I share that with you, hopefully you'll just see my own thought process, a window into my mind and my experience, rather than sitting here saying, do this, do this, do this, do this. Because who does when told? You only do what you feel like doing. So the idea is that you nurture talent and foster collaboration as a researcher. This is what I am, hardcore basic scientist. And this is the give to gain approach in science. My thought process in the last several years, since I've been an independent researcher and before that, has revolved around phosphate-rich biomolecules, polyphosphates, people, and partnerships, collaborations. So I'll elaborate on that. That's just photos I could dig out from my own childhood, my own ancestry. I come from a family embedded in the naval system, the Indian Navy. That there is my grandfather in his naval uniform. That's a portrait of my father standing next to his own portrait, wearing the naval uniform. In the military that I grew up in, the men went to sea, they went to office, and the women, the wives, stayed at home, looked after the kids, looked after the family. This was a very comfortable, very cushioned life. We did not know that people have different religions or classes. We only knew the men went to work and the women stayed at home. That's again from my childhood, as I said, very comfortable. My sister and I, we grew up around sailboats. We both learned to sail. And that's a very recent picture of the four of us, my parents, my sister and I, again, trying to get a boat into the water. My parents, my dad is now a veteran. Still there you can see a photograph of a ship behind him. We've been embedded in this, growing up around the sea and growing up with the idea that you serve the nation. That's my dad right there, 1967. passing out of the National Defense Academy, you can see only men. Things changed. You know when they changed? Anyone know when they changed? It's all on the next slide. So women were not allowed into the National Defense Academy when I was growing up. No girl growing up in the military ever thought she would be like her dad. I don't think I wanted to either, but it just wasn't even an option. Do you know when women were allowed into the National Defense Academy? 4 years ago. There was so much debate. My dad actually forwarded me some of the WhatsApp messages that all the men were exchanging as to how women belong or don't belong in the military. And last year, a wonderful thing happened. 2 women officers, Dilna and Rupa, Lieutenant Commander Dilna and Lieutenant Commander Rupa, circumnavigated the globe on a sailing vessel. They've completed their journey on the 29th of May. This is their picture from Cape Horn, the most difficult passage to sail. On the 14th of Feb, they sent us this pic. And 1 day later, the first batch of women on the 30th of May, 2025, the first batch of women graduated from the National Defense Academy. It has taken nearly 60 years of the National Defense Academy before women were allowed to join and graduate. Finally, we are there. Finally, the debate has ended. And in India, we've always sort of been behind the curve, right? Women have been accepted into combat roles in the armed forces outside India for many, many years. As a PhD student, I visited Israel. I went to Jerusalem for a meeting. There were women and young men there. Serving in the armed forces is compulsory there. Men and women in fatigues. Not here. Here it only happened last year. So a journey is always about making choices. And standing here before you was a chance for me to reflect on choices. As a kid there in my central school uniform, everyone sort of either took up engineering or biology, right? We really didn't know what else to do. And I figured, or medicine. Well, my grandfather really wanted me to be a doctor, and I was always a rebel. So I said, I'm not going to be a doctor. I don't know, something just didn't make sense to me there. So I took the engineering entrance exams. And in the meantime, I also went to Delhi University because my grandparents were based there and applied. My marks were not great, but I got into a microbiology course in Gargi College. And after that, in the meantime, I had taken engineering exams. I did my schooling in Vizag at the naval base there, and got into Osmania University here in Hyderabad in the computer science engineering program. And they sent that prospectus, which they would send by mail in those days. I got a little book with all the courses that you would take as an engineering student. And by that time, I had only bought this beautiful microbiology textbook by Pelczar with lovely color pictures. And I figured that black and white Xerox sheet of the prospectus of engineering college was no fun, and I decided to stay back. But it was a girls' college. In the meantime, I had written this exam in the All India Institute of Medical Sciences. They had a wonderful course called Bachelor's in Human Biology. And I got into that, and then I had to decide whether to leave Gargi College or to join AIIMS. And the choice was simple. AIIMS was co-ed, Gargi was girls' school, so I went for the co-ed. Makes sense, right? 17-year-old girl, she doesn't want to be with other girls. Okay. I then got into the Indian Institute of Science for my master's PhD integrated program and had to choose a lab. We were allowed to rotate and choose our own lab for our PhD. And I chose Sandhya Vishweshwaraya's lab for my PhD because she was young, cool, nice to talk to, doing interesting stuff. She's not so young, still very cool, still nice to talk to. And has been a major influence actually as a mentor in my life. I started working on cyclic GMP for my PhD. In the meantime, I met Raghu, started to hang out with him. That's the one occasion we were twinning there in the lab. He was in a neighboring lab. And towards the end of the PhD, we had to choose where to go next. Those days, everyone went PhD to postdoc. We really didn't know anything else. It's much better now. Happy to talk about that in a bit. I got into John Kurian's lab because he was visiting IISc at the time. He's a structural biologist. He asked to have a bunch of students talk to him, tell them about their work. At the end, he said, "Do you want to come to my lab?" So I went to his lab initially in New York. The lab moved to Berkeley, which was on the West Coast. In the meantime, we had gotten married. Raghu was on the East Coast. I was on the West Coast. It wasn't working. I then applied to Johns Hopkins, where Raghu was already. And I got into Saul Snyder's lab. And he was, again, one of the coolest person you will meet. You see here, that's him playing the guitar, singing carols at Christmas time. And very, very generous person. I learned generosity from him. He had 30 people in the lab. He remembered everyone's wife's, husband's name, children's name, where they were from. He couldn't remember all of it. He would write it down on that Palm Pilot we had those days. And then he would ask us. He wouldn't say, "How was your work? How was your result? Where is your data?" First thing he would ask about is your family. So from him, I learned to put people first. At the end of this postdoctoral tenure at Johns Hopkins, we kind of decided it's time to set up our own thing. We can do this. We've enjoyed working with younger people, setting up a lab, etc. So decided to become a PI. And also coming back to India was the only option by then, because I had this military training in me. It needed me to work and stay in my own country. Raghu and I agreed on that. And then we've been together here in Hyderabad ever since. He's in CCMB, I'm at CDFD. CDFD, as you know, has moved around a fair bit. And finally, we are within 5 kilometers of each other. So that's very convenient. Now, what I'll be talking about today are the polyphosphates we work on, the two varieties. One is very simple, polyphosphate. Okay, it's just a string of phosphates linked to each other by phosphoanhydride bonds. And then there are the inositol pyrophosphates. Now, inositol is a cyclohexane hexanol, 6-membered ring with 6 phosphates. Okay, and then IP7 has the 7th phosphate. इस पे ध्यान देना होता है यहाँ. And then polyphosphate. But more importantly, people. This is Manorama, our lab manager for the last 17 years. And without her, I would not be standing here today. No way. The backbone of the lab is the support staff and the people who really work behind the scenes. You will never invite Manorama here for Women's Day. But I can tell you that if not for her, our lab would not be where it is today. And also Dayanand, he's been working with us also for the last 17 years. When he started with us, his kids were really little. Now you can see they're as tall as me. And these two have really been the pillars of our lab. So I want to acknowledge them. Other partnerships. This is taken from Women's Day, where all the women in CDFD came and we just sort of spontaneously took a photograph some years ago. As I think this was we were just coming out of COVID. You can still see a lot of people wearing masks. It was just a spontaneous celebration, I think. And in here are some very, very brave women. Some brave women are in this photograph who have taken on the system and fought for their right. And they have received the support that they should have. Now that's everyone who has ever passed through our lab, all the people, each one with their own story, each from a different part of India, speaking a different language, following a different religion, philosophy, etc. But all of them have come through, and the ones in red are still here currently. And I've sort of tried to divide them into the 3 themes that we work on in the lab: the inositol pyrophosphates, polyphosphate, and rare diseases. I won't be talking about the rare diseases, but from the scientific viewpoint, if you want to discuss anything, I'm fine. We can talk about it later. These are our collaborations, past and ongoing. So these partnerships have been also the bedrock of everything we've been doing. In terms of, these are my collaborations with other labs within and outside of CDFD. Behind each lab, there are the support and the students who have also been our partners. Now, the theme in the lab is How are these molecules made? How are they regulated? What processes do they influence? How they work? मैं बहुत ज्यादा नहीं बताऊंगी इसके बारे में, बट जो लोग हैं, जो लोग हमारे साथ काम कर रहे हैं, उनके बारे में मैं ज्यादा बताऊंगी. When I was a postdoc in Saul Snyder's lab, along with Adolfo Sayardi, now at University College London, we uncovered a very unique post-translational modification. How proteins are modified by inositol pyrophosphates. So everyone's heard of protein kinases, and that they can transfer the phosphate from ATP onto a serine, threonine, tyrosine, histidine, aspartate, etc. So protein kinase CK2 can phosphorylate serines. We found that IP7 can lose this phosphate in the presence of magnesium, transfer it onto a phosphoserine to make a diphosphoserine. So we discovered protein pyrophosphorylation by inositol pyrophosphates happens in the presence of magnesium, but doesn't need an enzyme. Now, nobody believed us. We were trying to say that a PTM can happen without an enzyme. We met with a lot of skepticism. So when I started my own lab, we figured, let's start working on this modification and prove that we are right. So of course, we had to figure out how we detect it, how does it work, what cellular processes does it influence, how is it regulated. So instead of how does it happen without an enzyme, we decided to focus on what cellular processes does it influence. Now these slides look a little crowded, but that's because I've tried to tell the story of the person, not just the science. Now when Swarna, my first student, joined the lab, within her name she carried RNA. She actually wrote it like that, SWA small and then RNA big, and pasted it on her table, and she said, RNA work here, stay away. Because working in a regular lab, you need to be really careful when you're doing RNA work. Anyway, we had very little money at the time. We just started, so I chose yeast as a model system. It's inexpensive to work on. We had a wild type yeast and yeast lacking IP7. So one of the early experiments that Swarna did, this is when our lab was just set up, we'd gotten some equipment. That there is actually me. For the first 1 year, we were in an old building. I was in a borrowed office. You can see that my predecessor had the office super organized and gave me a little bit of space. And somehow we managed. And then we moved to another building in Nampally. Then I got my own office, I got my own space, and we got some equipment and we got started. And one of the early things that Swarna did is she went to Umesh Varshney's lab in IISc because he was able to look at the ribosome profiling. He had an equipment for that. And she worked with Srinivas Alluri, at the time her boyfriend. So that worked out very easily. So Srini helped her quite a bit run those experiments. And she found, if you can look carefully, this is in wild type, you have nice ribosomes, but very little in the knockout yeast that don't have IP7. To cut a long story short, what Swarna found is that IP7 transfers its phosphate to RNA polymerase 1, and then that is responsible for driving rRNA synthesis. So in the absence of or reduced rRNA synthesis, polysomes are less. Swarna went on to graduate and submitted her thesis in 2015. And I keep this slide because it looks so similar, right? That's me submitting my thesis in 2001. That's Sandhya, that's me, that's Swarna. The lab comes around when, you are doing something to mark the occasion and everyone's there in the pic. And I thought that was very cool. 14 years later, from my PhD, my student graduated. That's another pic of Swarna and her thesis. And in a couple of years, she became a mom. Of course, Srini, and she got married. And she had to take a good amount of time off to be a mother because her baby needed her at the time. And then she upskilled. She needed to get a work permit. They were in New York. It was tough for her. Took her a very long time. She took that time to upskill. She learned data science. She's now a data scientist at PepsiCo. Very, very far from the RNA she started with. She's and she tried explaining to me what she does. I don't even get it. All right. But she's doing really well. She's very happy at PepsiCo. And we met last year after a very long time. Manasa joined a few years later. By this time, I had, as I said, my own office. We got a very nice grant. We had lots of money, so we could now move from yeast to cell culture models. This is just an introduction to the enzyme pathway. That's the molecule we are working with, IP7. Its precursor, IP6, is made. This is made converted by IP6 kinases to IP7. So if you cross this out, ये कम हो जाता है. जब ये कम हो जाता है, then cells have a problem. All trafficking happens on microtubule motors. All right. Now, when you don't have completely functional motor called dynein, trafficking is a problem. So there's a traffic jam here. Oh, this went very fast. Sorry. Okay. And what happens here is the Golgi is dysfunctional. Transferrin doesn't traffic properly. And we found that's because IP7 pyrophosphorylates dynein. And when that doesn't happen in the absence of IP7, there's a problem. A few years later, another very important group joined our team at CDFD. We had Jayant, who's a colleague here. Very happy to see you here, Jayant, and Suman. And our director told us, go set up an animal facility somewhere. So we were in the middle of the city in Nampally. How many of you been to Mozamjai market? All right. Did you know there's no way you can set up an animal facility there, right? Been to Nampally, old city? Okay. So somewhere near the old city, we had a lab. And there was no way we could set up our animal facility there. So we went to Genome Valley. Every day we would travel 2.5 hours each way back and forth to the animal house. To set up, we rented a space there in Vimta Labs, and that's where we started our animal house. So by the time Ashik joined the lab, we had an animal facility thanks to these two, Jayant and Suman. And we started working on the IP6 kinase knockout mouse. And Ashik studied why the males are infertile. You can see here the spermatids are misshapen. So the males are sterile. Few years later, when Ashik was done, he got this very nice cover image in the Journal of Cell Science. As a biochemist, for me to see a microscopy picture was super cool. So that's how I felt that day. But something even more magical happened. Ashik and Manasa decided to extend their partnership beyond the lab. And then now they have a very cute 5-year-old whom I met last year. So you see how science and partnerships are interwoven. This is a diagram made by my student Akruti, just looking at all that IP7 does in the cell. And wherever these arrows are is what my different students have worked on. Of course, I won't tell you about all of them, but just to give an overview. Now, another very important partnership is a collaboration, a very long-standing collaboration with a lab in Germany. This lab is led by Dorothea Fiedler. Dorothea and I met when she was at Princeton, and she found that it would be interesting to work on this protein pyrophosphorylation. A common friend linked us, and then we've been working together for a very long time. She developed this mass spectrometry method to detect pyrophosphorylation. So if you are interested in technical stuff, just pay attention to this. What happens when IP7 transfers its phosphate to a serine to make pyrophosphorylation? If you take a phosphoserine and a pyrophosphorylation containing peptide, put it on the mass spec, what happens is this phosphate falls off and behind it leaves a dehydroalanine. So it loses a 98 Dalton mass. But when there's a diphosphate, it has 1 oxygen less. So it loses 178 Dalton mass. She looked at this signature, and by that she was able to identify 140 odd proteins. Around that time is when Dorothea wrote to us and she said, we've done the mass spec and they are a chemical biology group. Now I want to do biology. Let's work together and see if we can contribute something together. So Arpita, my student, had already been working on protein pyrophosphorylation of nucleolar proteins. So she took Dorothea's data and just examined it. She found that mostly pyrophosphorylation is on serine residues. They mostly have acidic residues close by. They're mostly disordered. And then, so this is Dorothea's postdoc, Jeremy, who had done that work. And then he left. And then Arpita and Leonie started to work together. Leonie from Dorothea's lab and Arpita. They communicated, email, WhatsApp, phone. Sometimes I used to tell her, please copy me on the email so that I also know what's going on, right? But in a sense, it's empowering for both of them, right? To feel that they don't need, they can work as equals anywhere with anyone in the world. You don't need your PI in the loop all the time. Of course, we kind of knew what they were doing and where they were. But they worked together beautifully. And this is the one experiment that took pyrophosphorylation from raised eyebrows, okay, to proof. So what they did is Arpita made proteins from cells that have IP7 and cells that don't have IP7. And then she shipped them across to Leonie, and Leonie ran them on the mass spec. And this is the pyrophosphorylation signature from wild type cells and the knockout cells. Clearly, these proteins, pyrophosphorylation is dependent on IP7. This is just, if, in case somebody is interested, this is the phosphorylation on these proteins, and these are the pyrophosphorylation, just on these acidic clusters. So we were able to put this work, which we did together, then went to Nature Chemical Biology, and now people have taken notice, right? We even got a news and news article. So finally, from the shadows, from disbelief, from saying you guys are working on something that doesn't make sense, it's probably an artifact, together we were able to solve the problem through this partnership between two women who are on the same wavelength. My last story is about Jayashree's work. When Jayashree joined the lab, we had just gotten, we were in the last stages of a Human Frontier Science Program grant. I'm happy to talk to you about this further. Please ask me about it, or please the faculty who are interested. This is a grant given only to do basic research, blue sky research, no application, no translation, and it is only given to international teams. So there was this team of us, including Paul Wender from Stanford and led by Henning Jessen from the University of Freiburg. Both of them are chemists, and myself. And we worked together where Henning made these polyphosphate synthetically. It turns out that if you heat phosphate, you can make chains of polyphosphate, but they won't be of defined chain length. They will be mixed chain length. To make a fixed chain length phosphates, beads on a string, okay, it required quite a chemical feat. So Henning made those, because that was a control. And when Jayashree did this pull down, she found a lot of mitochondrial proteins in there. Another very cool thing happened. Remember in the previous slide, I showed you about Arpita's photo? So Jayashree joined soon after. Can you see anything in this photo? On this, in the corner here, do you notice anything? ये देखिए, ये दो लड़कियां मेरे लैब में थी, और ये ध्यान से देखिए, बताइए कुछ दिख रहा है क्या आपको? बड़ा कर सकती हूँ मैं यहाँ से? No, I don't have control over it here. Can you see from there? No? कोई दिख रहा है familiar उसमें? ये दोनों यहाँ भी हैं. मिडिल में दिख गए? ये दिख गई अर्पिता? और जयश्री कहीं दिखी इनमें? कहाँ पे? फर्स्ट साइड में, यहाँ की यहाँ? ये ना? हाँ. ये दोनों पहले भी मिले थे कहीं और स्कूल में, अलग अलग स्कूलों से आए थे. ये बॉम्बे में कैंप हुआ था, पता नहीं कहाँ पे हुआ था शायद ये, और कुछ एक हफ्ते 2 हफ्ते के लिए स्कूल के कैंप में मिले थे, बाकी एक दूसरे को बिल्कुल जानते नहीं थे, वापस आके यहाँ मिले फिर से. So that's happenstance, coincidence, but I just thought it was so cool. So Jayashree then looked and found mitochondria have a lot of polyphosphate, and then she continued to work on that. That grant, the Human Frontier Science Program grant ended, and we got an Indo-German grant. This is around the time I found a new collaborator, Manish Jaiswal, who's very close to where NIAB is. He's at TFR, and he's a fly biologist. And both of us met at a university in University of Hyderabad at a meeting. And he had heard that we work on polyphosphate. So he said, is anything known about polyphosphate in flies? I want to pick up a project that nobody else is working on. And flies are a great system to do that. So I said, nobody is working on it. To my knowledge, nobody is going to work on it. So he's picked it up. And now he and his students, his group on polyphosphate is larger than mine. They have developed several fly models. They have their first paper out in eLife on flies as a models to study polyphosphate. They're making different lines, and I'm sure they will uncover metazoan polyphosphate biology more easily than we ever can using cell lines and mice. So Henning, Manish and I got this Indo-German grant. That is Sunana, Manish's first student on polyphosphate, Jayashree, my student on polyphosphate, two of us at a conference in, three of us at a conference in Spain. Henning's student came and spent 2 months with us, Sandra. Sandra Moser was thrilled at the prospect of coming and staying in India for 2 months. And she was the most friendly German I have ever met. Okay. She was like one of us. She literally, I mean, she didn't want to leave. And for my entire lab, it was such a nice experience to be interacting collegially with somebody from outside India, because most of them have never left India. Until you leave and until you work with people outside the country, somehow you have this barrier, this mental barrier about foreigners. And we've all grown up that way, right? It's all ingrained since childhood. But these kind of partnerships, they break those barriers. So Jayashree then did one very crucial experiment that in case some of the students are interested, I'll explain. She took liver mitochondria and added these electron transport chain substrates and found that they make polyphosphate. And when she added inhibitors of mitochondria, polyphosphate went down. So the other thing is that she picked up these IP6 kinase mice that Ashik had worked on, and she found that in the absence of IP6 kinase, polyphosphate goes down. She then went to Ullas's lab in TIFR twice, where his students, in his absence, because I think Ullas was already here at the time in TIFR Hyderabad, two of Ullas's students worked alongside Jayashree. They did a lot of experiments together, and our paper is going to be accepted. It's just been revised. And it has Manish and Henning and Ullas and their students and Jayashree and my other students. And all this was possible to discover polyphosphate function in mammalian mitochondria because of these partnerships. So Jayashree's work suggests that polyphosphate is synthesized by the ATP synthase in mitochondria. This was already suggested by others, but we have more proof now to cement that thought. Just a little collage on her journey. She started here with that Shyama Prasad Mukherjee Fellowship in the lab, and then went on to be a poly-P pioneer, in our lab at least, in mammalian mitochondria. She worked, as I said, in many collaborations, went to different conferences in CDFD, abroad, has a couple of papers, is also a badminton champion. You can see that here. Submitted her thesis. Her viva is going to happen this week. And more importantly, she's now a budding entrepreneur. She decided academia is not for her. She wants to be her own boss. And she wants to take up developing diagnostics thanks to being in CDFD. Did I say I did any diagnostics for her research? No. But just the atmosphere, the free interchange with other students, my collaborations with other colleagues at CDFD on genetic disorders, having that exposure and that atmosphere gave her the confidence that she can definitely do something unrelated to her PhD and make it her livelihood and start a business. So let's wish her all the best on that. She now is a BFI fellow at AIC. Just beyond my research, beyond what we do in the lab, beyond what we do in CDFD, there's so many other things to do. And one of them is India Bioscience. Can you raise your hand if you've heard of India Bioscience? All right, I'm so thrilled. Those of you who didn't raise your hands, please go look it up. We have been in business as an entity for quite some years now. Basically, it's a small office, primarily funded by a grant from DBT. And then we do our own fundraising elsewhere. It's an office of 5 to 6 people based out of the NCBS campus. And what our goal is to network researchers in the life sciences, right? We want to highlight life science research. We want to have young and not so young PIs get to know each other. And also, more importantly, we want MSc and PhD students to make use of what we can offer. So one of the things we have is a Crafting Your Career podcast. We also have Crafting Your Careers workshops. If you find a workshop, please enroll for it. We're now trying to restart them. Hopefully DBT will fund us soon. DBT funding is a whole pain point that, well, it's going to happen very, very soon this year. And this is a very nice resource book. So for those of you who may be master students or PhD students trying to figure out whether you want to be like your boss or not, this book is resourceful. So just a statistic, by the way, only 9% of PhD students are going to become academicians. 91% are not. So at least 91% of you need to read this book to figure out where your career path lies. And all that you have trained with us is training you for that future. It's not wasted. Everything you do, how you learn to speak, what you learn to think, all that applies to your career. And so this is just taken from Disha, all the, this is just, please open the book. These are all free online books on India Bioscience. The other thing that we've been working on is women in STEM. And I'm happy to talk more about it during the Q&A. SBC, the Society for Biological Chemists India, is a very, very old organization. 2 years ago, they did something radical. They had a panel discussion. They never did otherwise. At these SBC meetings, people would go, present their talks, present their posters, go home. They had a panel discussion on equity for women in STEM. Is India there yet? So we had I'd moderated that panel, and I think that was the beginning of more and more women researchers, students, postdocs, scientists, just starting to feel that we need as many conversations as we can. So there's another conversation, a group collective called Power Bio has started talking, women have started talking to each other, women scientists. We tied up with them, India Bioscience and Power Bio tied up with IIT Bombay. We had a workshop, 2-day workshop, dialogue on leadership roles for women in shaping academia. That is the logo. We had a very nice one there. And from that workshop has come an article written primarily by Sara Iqbal along with all of us who organized that workshop. And it has it's in press right now in a journal called Dialogue from the Indian Academy of Sciences. I encourage you to make a note of it. Please go through it, especially the young women here who are, and the men. Please, you are equal partners in this. It is on what young women researchers in different stages of their career feel. We had a very nice, round tables where we gave scenarios. We had people discuss scenarios. We had, Taru was one of the mentors for this workshop we had last year. And just all the conversations that came out of this are in the article. So please do go through it when it comes out. I had lots and lots of pictures. I initially clicked on pictures and I found 20 pictures. And then I said, cut, cut, cut. And finally, I got 16 here. This is just all that we do together as a lab, whether it's just sitting in the lawn for a birthday party or going out for lunch, going for a picnic, participating in foundation day, singing, dancing, etc. All of this, I feel, helps us just build that team spirit. We came up with 5 core team values: collegiality, enthusiasm, honesty, organization, and tolerance. And we selected this from 50 options. We all sat around the table, each of us came, we looked at these 50 possible options, and we selected these 5 top values and we distilled them. And we debated as to why we value these things in the lab. So this list, which the new students will see because it's pasted up there, it defines what we like to believe in as a team: helping each other. Honesty is just number 1. If you're not enthusiastic about what you're doing, just what's the point? जोश होना चाहिए, एक दूसरे से दोस्ती करनी चाहिए, हमेशा सच बोलना चाहिए, राइट? झूठ बोल के किसी को कुछ नहीं मिलेगा, और अपना काम ठीक से करें, दूसरे की परवाह करके अपना काम करें, दैट इज ऑर्गेनाइजेशन. और दूसरे को अपने से अलग ना समझें, और अगर कोई अलग है तो उसको डोंट अदर देम, दूसरा ना करें उनको, इन द सेंस की टॉलरेंस. हम सब इकट्ठा काम कर रहे हैं, हम सब एक साथ काम कर रहे हैं, आपका काम भी इंपॉर्टेंट है, मेरा काम भी इंपॉर्टेंट है, कोई किसी से ऊपर नीचे नहीं है. ये सब चीजें हमने सोची इनके बारे में, और इनको हमने लिखा ऐसे यहाँ पे, क्योंकि और अपने सामने रख के आगे बढ़ते हैं. So this was the only time that I thought, okay, now give to gain, Madhuri. I needed to reflect. So I'm a biochemist. I figured after making this presentation together, I said, okay, what is my role? What am I? So I said, I'm an enzyme. I feel like I'm an enzyme, right? Because what does an enzyme do? You don't have to do very much. You just have to stay in one place and let the substrate and the, become the product. You just have to be just catalyze stuff from as it happens. Right? You don't have to change who you are. You just have to facilitate things as they happen. So I just came up with this early this morning, and I typed this here. Okay, I'm an enzyme. The give to gain thing. What have we given? What have I described to you as the giving in the story? Mentorship, time, empathy. There are enough instances where people don't get along, people struggle, right? What do they need? They don't need to be told what to do. They just need a listening ear, and they need to be told that it's okay, we'll figure it out. You don't have to leave the lab because you have a terrible problem at home, right? We'll work through it. We'll make things happen here for you. Come back when you're ready. That kind of empathy is, I think, very important. Balancing that with rigor. There is no chalta hai at it, no room for chalta hai. Kuch chalta nahi hai. Jo karna hai, perfectly karna hai. Aapka har experiment needs to be perfect. How to stand up and talk. जब हमारे पास आप लोग आते हैं, how many of you can stand up here and give a 1-hour lecture the day you arrive in NIAB? Anyone? When you're ready to leave NIAB, how many of you stood here and given a 1-hour lecture on your work? Come on, half hour talk, work presentation in auditorium. Yeah, only 5 of you. Everyone's in first year. Otherwise, lab meeting, work presentation, colloquium, right? Journal club, right? Just the courage to come and stand up here. अपने आप नहीं हो जाता ना, help चाहिए होती है, right? यहाँ खड़े होके to talk about even your work. That training, that happens how? अपने आप हो जाता है? Somebody is helping you, right? Your lab mates, your guide, your other peers. They give you the incentive. They tell you you can do it. They help you practice. And then when you do it, the more you do it, the more you learn. Scientific writing. In the days before ChatGPT, even in the days of ChatGPT, writing is a skill you have to know, right? Most importantly, ethical research practice. So we give a course every year on research and publication ethics, and that helps us learn. It helps us reinforce what is okay, what is not okay in research. Collaboration, I talked about it. Supporting younger colleagues. We must never forget where we were when we started, especially younger women colleagues. It is a fact, not a myth, that women have something called imposter syndrome. Have you heard of that? What is imposter syndrome? Anyone? I'm not good enough for this. It is very common, it is documented that women tend to think, nahi, ye mere liye, main nahi kar sakungi, ye mere se nahi hoga, koi aur kar lega, lekin I am not good enough. Whereas men don't have that. Men feel, कूद के देखते हैं, हो जाएगा कुछ ना कुछ. हम लोग ऐसा जाएं कि ना जाएं इसके अंदर, right? So that is a fact, and that holds women back. If you recognize imposter syndrome and see it in yourself, and then try and work with others when you see it in them. So every year I go to the young investigators meeting where I meet young PIs who are just starting. I just came back from there last week. And those who want to apply for academic jobs, I can see the difference. The men are, I am so and so, and I want this question. And the women are like, पूछे कि ना पूछे? They come and say, आपने हमारा question पूछ लिया, thank you. Like, why didn't you ask? I don't know if I'm ready. Shall I go and talk to that person? Nahi. So one young woman came to me and she really, I said, you should go talk to Ullas. He's the right person for this. No, he's busy right now. He's talking to somebody else. I said, have you talked to him? No, he's eating lunch. I will find him later. Finally, she did find you. But I had to tell her, it's okay. You're here for this. Don't hesitate. Right? I'm sure none of that, I saw it. Most of the men didn't feel that way. They felt ready to take that step. Somehow we are hardwired. And it's not a myth. I didn't make this up standing here. It is documented, right? So if we don't support younger women colleagues, men and women don't recognize this and don't support our younger women colleagues, this cycle will continue. The cycle will perpetuate. Entrance exam देके आप लोग जो यहाँ तक आए हो, उसके बाद there is no more entrance exam. The next part of your career is there for you to take, for you to say that I can do this, right? Nobody is going to offer it to you on a platter. You have to take it. You have to feel confident to get there with support from your senior colleagues. Of course, we've also done student and staff welfare, institutional responsibilities, and contributed to the international and Indian research ecosystem. What have I gained? New discoveries. Our science has been recognized nationally and internationally, but more important, there's been a ripple effect. Each person I mentor, each of my mentees now becomes a mentor for somebody else. Initially within the lab, later on in their next stages of their career. And this legacy of mentoring young people lasts longer, much beyond publications. Seeing my mentees shine is what makes me feel that I have gained. And of course, the reason we are here, the reason we didn't stay back where we were in the US, we are contributing to the nation's progress. We are contributing through our science and through our mentoring of all the young people here. So lastly, Samgyan. This is a photo I took when I went to Sabarmati Ashram. It's there on a pillar. Be the change you wish to see. You can be the enzyme, just catalyze the change. But actually do something. Don't ever complain about how things are. This was on the back of a passing car, and I thought it was so appropriate for the theme today. Nobody has become poor from giving. कोई देने से गरीब नहीं होता है. देने से बढ़ता है. You get it back. Whatever you give, you get back. And this I really like. I usually tell this to college kids, but I think it applies to PhD students as well. Right? It's my favorite quote from Baba Ranchhod Das. Chase excellence. Success के पीछे मत भागो, काबिल बनो. कामयाबी झक मार के पीछे आएगी आपके. And then what I felt again through pictures conveys how we should all be. We are in this together. So there's this nice little selfie stand. The day it was put there at CDFD, 3 of us took a pic. So that's me with Amrita and Begum. And again, I've been in CDFD very long, as you can see, but the excitement shouldn't go away, right? People before papers always, if we look after the people, the papers will come with them, right? We are celebrating Jairaj's thesis defense that day. And celebrate the small joys. This actually, do you recognize anyone in this on the stage? Ullas. Where is he, front or back? Back. So Ullas, I wanted to put this in just to symbolize how we all feel as a community, what partnership we feel. This is the Accounts and Finance Department receiving their Best Section Award just literally about a month and a half ago. And they said, we want to dance, but we are not sure. I said, Karo, kuch nahi hoga. Karo dance. So then they had this fireworks, and then they're all dancing. After a while, they forgot their director was behind them. They were having such a good time, right? So that was a small joy, right? Why not? I mean, there were about 150 people there. They were all happily dancing. Because if you don't feel that joy in what you do, that reward in what you do, वो रंगोली जो आपने बनाई है, वो खुशी से बनाई है ना, मन से बनाई है वो रंगोली आज के लिए. Those little things, if you don't celebrate, then what's the point? So that's just my customary acknowledgement slide. So I did talk about our science as well, and the facilities that we have, all my colleagues, all collaborators, that's what CDFD looks like. It's just a very wonderful place to work. We're very collegial. We're small. We don't have to walk around campus very much. Just go up and down. Everything is open. Walk in anywhere you want. And just happy to share this wonderful day with all of you. And thank you for being here.
Thank you so much.
Sure.
Just say that those students bring your talk today, they have something big.
Thank you.
Now the floor is open for questions.
Yes, I'd love to talk about Women's Day, what it means, anything else.
Maybe from here. Who wants to go first?
All right, otherwise I'll start a quiz or something.
Yeah, you can do that.
Yes, Harjeet ji.
Thank you, ma'am. It's really wonderful.
Thank you.
I was expecting some turbulence in the talk.
Some?
But ये तो कुछ turbulence आएगा, कुछ problem आएगा.
Problem
ये
नहीं
तो
आई.
problem in your life and how you handled.
Okay.
ये तो सन्नी देओल पाकिस्तान गया और नलका उखाड़ के ले आया.
ये तो कल की मैच की तरह हो गया, no contest.
What we wanted to know कि एक ऐसा tough time
जी.
अगर आया,
हाँ.
and then how you handle that.
जी. देखिए टफ टाइम तो आता ही है, बट वो थोड़ा आपके एटीट्यूड पे डिपेंड होता है. एक टफ टाइम जब मुझे आया तो मैंने सीनियर कॉलीग से बात की उसके बारे में कि देखिए ऐसा ऐसा हो रहा है मेरे साथ. मैं हैंडल कर लेती हूँ, मैं बहुत कॉन्फिडेंट हूँ, मुझे कोई कुछ नहीं पीछे नहीं कर सकता. एक टाइम था जब थोड़ा मुझे मुश्किल होती थी कि किसी ने कुछ बोल दिया तो मैं थोड़ा नाजुक सी थी, आंसू निकल जाते थे. तो थोड़ा फिर टफ हो गई, क्योंकि बात ये है कि आप अपने काम, यू टॉक थ्रू योर वर्क. अगर आपका काम अच्छा है और लोग अब आपके साथ हैं तो कोई आपका कुछ बिगाड़ नहीं सकता। लेकिन एक समय था ऐसा, तो फिर मैंने अपनी सीनियर कॉलीग से, एक हमारे इंस्टीट्यूट में नहीं कहीं और, उनसे जाके सलाह ली। तो उन्होंने कहा देखो आप हैंडल नहीं कर सकते क्या ये? मैंने कहा बिल्कुल हैंडल कर सकते, मुझे कुछ कंप्लेंट डालने की जरूरत नहीं है, मैं हैंडल कर लूंगी अपने तरीके से। तो देन शी सेड डोंट वेस्ट योर टाइम, आप हैंडल करो, कोई क्या कर लेगा आपको। जब कोई और हैंडल ना कर पाए तब आप उनका सहारा दो, राइट? Help those who need your help. You don't need it. You can manage. You can tell people you don't care if they don't, if they're not doing the right thing. So I did actually do that instead. एक मौका ऐसा आया जब मैंने फिर किसी और की सहायता की जो अपना नहीं कर पाई थी, और उससे मुझे लगता है कि my toughness, I may seem very super nice, but can also be tough, right?
By
So
the way, my senior in IAC, and they were all used to be extremely scared of that.
so I think that's something that's not really easy, and a lot of people struggle with it. That how are you going to be rigorous and also nice? But somehow I figured that out. Like no nonsense is going to happen when I'm around. Yet, if you are right, I will stand by you no matter what. So I did stand by somebody no matter what. And that was a tough time for all of us, but we figured it out.
Yeah, thanks. Very nice. I really like your tips on mentoring.
Yes.
I think our students, I wish they recognized and realized what you said.
Yes.
And any more tips that you can give on good mentoring and being good mentees and mentors?
Certainly, I'm doing that for a living right now. In that sense that every year we go to IIM, I'm just fresh off the latest one. So for mentorship, I have a few stock phrases. There is no one size fits all. So when we start, the last thing we are trained on in our PhDs is how to be mentors beyond teaching our junior colleagues how to do the best experiment. That you can all do, right? By the time you're in third year, fourth year, you have a first year student coming, how to do a perfect experiment, you can teach them. That is not mentoring, right? That is only teaching them the experimental rigor. What we are never taught is how to be a mentor. That we have to learn. Some of us do a better job than others. Some of us try really hard and get better, dhakke kha kha ke, right? But one thing that we've learned is there's no one size fits all. Each of your mentees, you have to tailor make your style for. There are boundaries, there are lines that will never be crossed. Honesty is never going to be compromised, right? But how much time each person needs from you, what level of nudging, hand holding / independence they need from you, who needs what, who needs writing training, who needs presentation, who needs to be told don't be afraid, who needs to be told कूदना बंद करो थोड़ा, सोचो ज़्यादा, those things you will need to think about each person. And the biggest mistake we tend to make is we tend to self-project. We tend to think that everyone thinks like us. Actually, that's also not true, right? Each person has a different thought process, and we try to see them in our mold. Sometimes we are surprised, अरे मैंने तो कभी सोचा ही नहीं, उसने ऐसे कैसे सोच लिया? But I think recognizing that. So no one size fits all, and just tailor making your mentoring to each mentee is the most important thing. The rest will follow.
When compared to 2 decades ago, you see a change in women and students going forward. Are we stuck? And I feel we are stuck, but I could be wrong. But I would like to believe we have moved.
In India, we are still very far. In India, I think we are stuck. There were a good number of women students at my time. The faculty ratio was abysmal. It continues to be so. In India today, we are 16% women faculty across STEM. That's a recent study has been done. All over the world, we are 33% women in all levels in STEM. In the life sciences, we have crossed 50%, right, at the entry level. Something is seriously wrong, and it has to do with attitudes, physiology, and systems. Attitudes of women towards their own thought process, attitudes of the system, right? It's just, it's not, nobody's trying to pull anyone down. What are faculty saying who are in recruitment? Applications ही नहीं आ रहे. What are women saying who want to apply? Am I good enough? I've taken a career break. How many hiring systems give any age relaxation for a career break? What's the age by which you're hired into faculty? Again, I said I'm come fresh from YIM. The average age in which we like to recruit our faculty is 35. In the life sciences, you barely finish your PhD by the time you're 32, right? How many here final year PhD students? Yeah, 32, 33 sometimes, 35 sometimes, right? Because you took a 2-year break trying to prepare for that CSR exam. And then you have to do a long postdoc, long PhD to publish a paper, long postdoc. Where is the relaxation in the hiring age in the life sciences? None. The BRIC system, the DBT system actually has given us some relaxation. They'll take entry level up to 40. But we retire at 60. Who wants to hire 40-year-olds? Right? Everyone's trying to hire 37-year-olds. Which women who have wanted to have a child or 2 children for that matter have enough on their career track, enough good publications by the time they're 37 in your age limit? You know, Shekhar Mande tried very, very hard to bring that CSIR 35-year age limit in hiring to make it up to 37 or 40 for women. He failed. Tried for 3 years as DG. It's not that people are not aware of the problem. It's just that the systems are so entrenched, right? So then a lot of women, biologically, this is the time that you have to finish your PhD, get into a postdoc, start working towards building your academic credentials, are also the time that you have to then choose, do I start a family or not start a family, right? I have so many male colleagues, so many of them. Their wives are homemakers. Very few of their wives are not homemakers and have an independent career. Why is that? It's the same system I grew up in. The men went to office and the women stayed at home. I know so many women who did PhD along with me and have decided that their science ended with their PhD. Systems are not okay. It's not family support. It's, in fact, in India, it's much better. Yes, daycare helps if you have it on campus, but so many people have other help. Again, at YIM, I met 50% women faculty, all of them, like super inspiring. One of them had come with 2 kids. I said, did you bring any help? So we give childcare grants. So I said, did you bring any help? You've got 2 kids with you. She said, yeah, my husband, he's here in the hotel room. That's what we need, right? Which man will think twice before bringing his whole family, putting them in the hotel room and going for a conference? I didn't even think to ask if her husband was looking after the kids. My bias. I thought it'll be a mother. So that's the change. It's coming, but it's very, very, very slow. We all need to change our attitudes. Men have to be equal partners. How long is paternity leave in India? Anyone? 15 days. You wish it were 3 months. How long is childcare leave for women in government job? Huh? 2 years. See, admin people know this. What do our colleagues feel when they are hiring women in permanent jobs?
I wanted to take childcare leave,
Mila nahi na?
but admin said men are not eligible for
Not
childcare
eligible.
leave.
15 दिन, baby born, जाओ wife को पंखा झेलो, जाओ काम पे. नहीं है. It's not in the system. क्या कर रहे हैं हम उसके लिए? कौन झगड़ रहा है उसके लिए? Are our male colleagues fighting for childcare leave? Have they gone to rock the doors of government for childcare leave? No. Women, and then if anything in government, if you can take 2 years of childcare leave for women, it's actually a negative mark on the hiring. इसको लेंगे तो ये तो दो साल घर पे बैठ जाएगी. That is, it is said. ये तो उसके बच्चे तो छोटे हैं तो काम पे ही नहीं आएगी. ये भी मैंने सुना है. I'm not making this up. Right? So childcare leave was introduced around 2009, I think, 9 or 10 for women, right? 2 years in blocks of 3 weeks or 1 month or something like that, or 15-day blocks, something like that. It used to be, you had to take a minimum of 15 days. Now I think they've changed that. So there are entrenched issues, but it starts with us here, right? It's not straightforward, but if we don't do it, who will, right? So all of you young women here, do something about it. Take what is yours to take and find willing partners to help you in the journey. It'll change. I'm always glass half full, but it has changed. So I put this question to my friends who are in the US, most of them, and who did college together, and many of them are in academia there. They have 40 to 50% women professors there. Something has changed. Right? When I was in the US as a postdoc, 40% of assistant professors were women, 20% of full professors were women. Those 40% assistant professors are now full professors. It has changed. There are more and more women in leadership roles. They have more and more women in the faculty. They figured out quite a bit. We are yet to get there. Yeah.
Ma'am, so we all have realized the big gap between the number of students who join a PhD versus the number of faculties that we find in women. So as students, what can we actually do? I believe there would be something that is happening also at the PhD level, that our colleagues who actually are working with us, maybe we are not creating an environment enough that they feel supported to go ahead. So what could we actually do at the student level to support our colleagues?
I think just first you start off by eliminating any bias. Like, I'll give you a simple example. If somebody is saying, when the student comes and when we hire him, he will do this course, and then what did I say wrong? Huh? Stop that. There's an alternative. Just say they. Just become gender neutral. Start there. He is not a gender neutral pronoun. Manpower is not a gender neutral term. Let's just stop using these terms, right? That itself tells people everything is level. It's just a start. It's just a little mindset, right? When the time comes and your female colleague in the lab is deciding where to go for a postdoc, and she wants a sounding board to talk to you and say, यहाँ जाऊं क्या, मैं क्या करूं अभी, apply करूं कि ना करूं, find something and tell her to apply, right? Say, take the leap and figure it out later. Don't overthink it, right? Just be there. Just be that support. When you find a partner, support your partner as though your partner would support you. The gender roles are reversed. Let's hope for the best. I'm really exceeding my time now. Is this the Q&A? Yeah.
What keeps you going on? Like, there are days you feel time.
Of course. There are days when I feel fatigue of everything. Not so much the science. I wish I had more time for the science. I meet my students after 6 p.m. some days. There are so many other things going on. But science is what the passion that brought me here in the first place. And I think you need that passion, right, to do a PhD, first of all. It's the hardest thing you'll ever do, that PhD, right? It's just not for the faint-hearted. It's really, you need that kind of passion to keep going. And it's not there every day. Some days you'll just be like, chuck this, yeah, let's do something else. But you need to keep that going.
Balancing relationship and PhD.
Oh, yeah. So that's the other thing, right? That's the time for so many women that you're going to make a choice of your partner. And many families are saying, "Bhai, kitni lambi chalegi PhD hai? Umar ja rahi hai bachcha paida karne ki," right? So, I mean, there's all of that, but you have to make everyone your ally, right? You can't make everyone your enemy. You can't fight with your family. They have to be your allies. So it's tougher. Huh? It's not easy. There's so many things. How many boys are being asked, कितना लंबा चलेगा PhD, बच्चे पैदा करने का time जा रहा है. Yes.
Ma'am, since you mentioned about lots of mentorship that you've provided to your students, and you being such an amazing guide with your peers, I would like to know what would you suggest since women reproductive health or women research based on women's health is already a big lack, I would say, all over.
Completely. Yeah.
So we as women already have to put in like 2 times the efforts to reach where men are in general, and then focusing on a field like women's health will be like 4 times the efforts because it's already lacking. So what do you suggest as a mentor, like to a student who has not even started the PhD, looking for it or planning for it? So I would like to get a, I don't know, highlight of what I can expect and how I should move forward.
So your question is that what is the scope for research in women's health?
Yeah, and looking forward to the what do you call it, the hurdles that is going to be there, given it's there. So how should you guide your mentee in that?
Okay. So two things. It is true, and it has been recognized a little while ago that research in women's health is lacking, like far, far behind. In the US, for example, they realized that most trials and everything was geared toward, they're called WASPs, WASP, white Anglo-Saxon Protestant males. Okay. And they realized that a little while ago. Of course, now that things have gone the other way around, where DEI has been trashed and all research in, whether you're focusing on women or other genders, or whether you're even focusing on other ethnic groups in the US, it's all gone for a toss, and it's going to take decades to come back. But we don't have to go that way. We need to at least realize from that. And therefore, a lot of effort went in a few years ago to emphasize that, for example, when you do studies in mice, when I was doing that study as a postdoctoral fellow, everyone only said, no, only do with male mice, otherwise the estrous cycle will mess up your data. Right? So now, if you try and publish some work in mice, you have to show that you've done the work in male and female mice. Forget the president of the US who doesn't care about your mouse experiment. Your colleagues who will peer review your paper will not let you publish work only done on male mice anymore. So that realization has come. Having said that, if you just do your own research, I mean, as a woman, you go through everything, right? You go through your periods, you go through childbirth. Well, I didn't have kids, but you do, and then you go through menopause, and at each stage, it's like, इसके ऊपर पता ही नहीं है, इसके लिए कोई दवाई नहीं है क्या? Right? So, and then everywhere you try and read it, it says little is known about this. Why is little known about this? Right? So more definitely needs to be done. And women don't even talk openly about these things. At least your generation talks openly about their periods, right? My generation didn't. It was taboo, right? So at least you need to start there. How can you help your female colleague if she can't come to work on the one day of the month? At least, run her gel for her, set up her Western blot. And don't make her uncomfortable about it. Right? Normalize the subject. So much needs to be done about women health. You are in the right place, go for it. You will, in the right climate, you will get funded also. You had a question?
So I don't have a question actually. I am a mother of 15-month-old baby. I'm doing my postdoc here. So as soon as I finished my PhD, I delivered. Then I took a 1 year break. So having highest degree in hand and sitting at home, that frustration was.
I've heard this. Yes.
Yes. But then now, okay, I started with my postdoc. Again, only both of us are here, me and my son. So research is very demanding job. Once I go back, I have to take care of him, the home, the cooking, cleaning. Like my day starts after he sleeps. Next day
Yeah.
morning, every day I have to be present here. The whole day is going with the work. So maybe this, maybe sometimes I don't, I am not able to take it. No, I just want to quit. Maybe this is the reason that there is no women in science. Like,
Absolutely. This is the reason that women don't go beyond PhD and postdoc, because they are torn. They're torn about the guilt, right? Physically and emotionally, it's the toughest thing to do, to leave a baby at home and come here and do some work which you don't even, at the end of the day, like, kaun sa tir maar liya maine experiment karke, right? You get that feeling, na? So it's the hardest thing to do, and you need so much support to be able to do that, and so much strength of mind to be able to do that.
you start an experiment, you have to decide, okay, am I finishing it by 5? If not, baby will be, okay, the daycare is only till 6. I have to finish this. How I should finish?
I don't know. She said you have to work twice, 4 times as hard, right? You can't take a break. You can't. So I had a postdoc in the lab who had a young girl at the time. I think her daughter was 5. Your kid is much younger. And she's the one who worked smartest in the lab. Everyone was like, 11 बजे से 11:30 चाय ब्रेक, 1 से 2 लंच ब्रेक, 4 से 4:30 चाय ब्रेक, and she was like, come 9:30, leave at 6. And everyone used to say, कितना काम कर लेती है वो एक दिन में, हमसे तो होता ही नहीं, right? Because you have that motivation, that mindset. So you need allies, and you need to be able to talk about it, because then if you talk about it, those allies are going to tell you your kid is not going to be a baby forever. But if you stop now, then you may not be able to get back. Yeah. You have allies. You're standing up here and talking about it means you have allies, because you're comfortable saying this in front of them. So they are your allies. Thanks for sharing that. It's time to finish.
Thank you, ma'am.
Thank you
for
so
such a
much.
wonderful Thank you so
Thank
much. It was very insightful. Now I do humbly request our director sir to please felicitate him. Okay. Please, both of us.
I will be there.
Okay.
Do you want? Okay.
I think you should.
Oh, nice.
I can say.
What are you, young? Okay, I'm old. That means I'm old. I don't like to be called old.
You are right.
Thank you. Thank you. Oh, wow, this is beautiful. Thank you so much. Thanks.
So we also take this opportunity to thank and acknowledge the efforts of the wonderful women of housekeeping team. ఈరోజు మన బ్రిక్ ఎన్ఐబి హౌస్ కీపింగ్ టీం మహిళానికి మా హృదయపూర్వక ధన్యవాదాలు. మీ కష్టపావు వల్లనే మన క్యాంపస్ క్లీన్ గా అందగ్గా ఉంటుంది, మరియు మన ఇన్స్టిట్యూట్ స్వచ్ఛత అవార్డ్స్ కాదు వస్తున్నాయి. దట్స్ ద బెస్ట్ టైం ట్రైయింగ్. ఇప్పుడు దయచేసి స్టేజ్ మీదకి రండి. ఎన్ఐబి నుండి ఒక చిన్న అప్రిసియేషన్ తీసుకుదానికి. థ్యాంక్ యూ. ప్లీజ్ కమ్. I will take the name. Please come. I would also request Rachna ma'am to please join, sir, and Harjeet sir to please do the honors. Please. Nagmani, please give a round of applause to them, please. Thank you. Nagmani, Menka.
Nagmani. Thank you. एक फोटो लेना है ना? आओ जाओ फोटो लेके थैंक यू
मेनका, रूपवती, Sunita, Adi Lakshmi, Sharada, and Sudha Rani ji. Please come near the stage. Rupavati,
नमस्ते रूपति
Sunitha, Adi Lakshmi,
नमस्ते आदि लक्ष्मी
Sharada,
नमस्ते शारदा
and Sudha Rani.
सुधारानी
Sudha Rani.
सुधारानी नमस्ते सुधारानी
Thank you, sir. Thank you, ma'am. Thank you.
थैंक्स
Acha, one group photo.
Photo?
They are requesting for a group photo. Please come, please.
Recently lost her husband, but Supavati,
she couldn't come today
she was here,
okay
and she feels coming to work is actually, you know,
the
giving
way
her
that she's
happiness
able
by
to
herself.
manage very good
So I think NAB is enabling that.
very good it's very
And
this I think we need that off no the projector or we need it off It's okay. Come in front thank you
Thank you, sir. Thank you, ma'am. It means a lot.
can I just turn this off I realize it